Now we're going to talk about the transition step, and the transition step is just a short kind of pathway that takes from pyruvate to acetyl-CoA, and acetyl-CoA is the start of the Krebs cycle. So this is just kind of an intermediate to get to that pyruvate to acetyl-CoA, which is why it's called a transition step. There are 3 enzymes that work together to do this one step, and that's going to form a complex that we'll learn about here in a second. But let's start right where we left off with pyruvate. So, as mentioned earlier, this is going to take it straight to acetyl-CoA. Now, the 3 enzymes that allow this to happen are pyruvate dehydrogenase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase. So those 3, I'll write them all out. And something kind of special about these 3 enzymes is that they all use a kind of cofactor. So for pyruvate dehydrogenase, it needs TPP to function. For dihydrolipoyl transacetylase, it needs lactic acid. And for dihydrolipoyl dehydrogenase, it needs FAD. Now, this entire complex here is also called the pyruvate dehydrogenase complex. Also, in this reaction, we are going to be losing carbon dioxide. Also, in this step, we are going to be reducing NAD to NADH. So, if we're keeping track of our carbons, we know that pyruvate has 3 carbons because it was that 6-carbon glucose that got split into 2. So pyruvate has 3 carbons, and we're losing a CO2, so that takes us down to 2 carbons. So acetyl-CoA is only going to be a 2-carbon molecule. Sometimes it's easy to count the carbons if you get mixed up in the steps, but this is all there is for the transition step. I'm going to label it real quick. And that is all that there is for this transition step. It is just those 3 enzymes that form that one complex that just take it from pyruvate to acetyl-CoA to kind of kickstart the Krebs cycle. So we're going to do the Krebs cycle in the next video, but the notes from this will also always be in the link tree, and it will be under Biochemistry transition step.